All right, so today here on campus we did an enrichment, and what we did were these two problems, which you can download from the course website, or you can just pause the recording and then try it for yourself and then resume when you're ready. So here's problem number 1. Segment of protein was given, and I want mRNA and DNA. And then here's number 2. You want a polypeptide to have this sequence. So then give the mRNA and DNA, give a DNA with silent mutations. Could this same polypeptide be produced with insertion and deletion mutations? All right, so now we're going to go over these problems. Make sure everybody understands how to do problems like this. Now you're going to need your handout with your structures and your codes. So how would you go backwards, right? On the last enrichment, I gave you DNA and you turned it into this. So how do you go the other direction? What do you have to do first?
You divide each peptide up.
Okay, so you break it up into the amino acids. That's what you did first? Okay, so let's break all the peptide bonds. Okay, then what'd you do?
And then you find each one of them.
Okay, so you identify each amino acid, right? And you do that by looking at the R groups, right? So what amino acid is this? Alanine. And then what's this one? Okay. And then what's this one? And this one. Right, so break it up into its amino acids and identify them, looking at the R groups. And then what did you do?
I found the ALA on the other side, and then I took one of the codes to make the RNA shape.
Okay, so you then made an mRNA from that. Now, notice the question says, state acceptable. Why is it acceptable? Why did I use that word? Because are there more than one codons for alanine?
Yeah.
Right, so you could have listed either one. So if we find alanine on our list, ALA. Okay, so we've got 4 choices here, right? GCU, GCC, GCA, GCG. All 4 of those would be acceptable. Which one did you pick? GCC? Okay, that works. GCC works. But you also could have written GCU, GCA, or GCG. Okay, now we find GLU. What did you pick? There are only 2 to choose from there, right? GAA and GAG. So GAA, okay, that would work. And then for cysteine, let's see, we've got 2 choices. UGU or UGC, which one did you pick? UGU, okay. But UGC would have also worked. And then for glycine, let's see how many choices we have. We've got 4 to pick from. GGU, GGC, GGA, GGG. Big GGU, all right. So do you understand why you could have picked any one of those 4, right? Because any one of those 4 would have produced that same amino acid. And then how did you get from mRNA back to DNA?
I did the complement.
Right. So you just write the complement. All right, so G would be C, G, G, then C, T, T, then A, C, A, then C, C, A. Do we agree based on that mRNA sequence? Now again, remember, there are multiple mRNAs that would have given this. Would they be the same DNA though? If you picked, instead of CCC here, if you picked, for alanine, GCC, if you had picked GCG, right, GCG would have also worked, but that would have been a different DNA, right? That would have been C, G, what did I say? C, right? So, as long as you understand the method and that there are multiple ones that would work, that would work. So you understand the process of going from this back to the DNA. That makes sense? All right. Let's go now to number 2. All right, you want the peptide, polypeptide to have this sequence. So again, we need to go through the same procedure that we just did. Okay, we need to first break up our amino acid, break up our polypeptide into its amino acids. Using our handout, what's this one? Phenylalanine, okay. And what about this one? Threonine. And what about this one? Aspartic acid. And this one? Valine. Okay. Now again, I said give me acceptable, which means there's more than one that would work, right? So let's go through the process of turning this back into mRNA. So, phenylalanine, there are 2 choices. I'll just pick U. For threonine, there are 4 choices. I'm going to pick ACA. For aspartic acid, let's see, there are 2 choices. I'm going to pick GAU. For valine, there are 4 choices. I'm going to pick GUG. Right, so as long as you pick an mRNA that codes for each one of these, even if they're different than the one that I happen to pick, right, so long as it's one of the acceptable ones, especially not stop, right, then you're fine. Then we just turn that back into DNA. So if I've got U, what would the DNA be? A, and then what? T, G, T, C, T, A, C, A, C. Good. How did yours differ from mine?
The only one that we had the same was the first one.
Oh, really? Okay. But we would have ended up with this exact same polypeptide. Okay, so I'm going to leave my DNA up because part B asks, give a DNA sequence with silent mutations on nucleotides 5 and 8 that would still produce the same polypeptide. So what is a silent mutation?
It's a mutation that you change one of the letters, but it doesn't change.
So what kind of mutation is it? Point, insertion, or deletion? It'd be a point mutation, right? It's a point mutation. So is the DNA different? The DNA is genetically different, right? You are a mutant, so to say. But what happens in a silent mutation?
The change isn't really apparent.
Right, there's no effect in protein. Okay, so if I want silent mutations on 5 and 8, 1, 2, 3, 4, 5, 6, 7, 8. All right, so that, let's just go ahead and break this up into our triplets. How do you pick what to turn it into?
You have to go back to that protein and see what else there is.
Right, so what are our other options? And I forgot to write the sequence.
Is that like a trick? Because you can't change the second one, can you? Aren't they all the same? The last one's the one that changes. right?
Yeah. So let's go back. What were the names again? I forgot that.
PHE, THI, AST, and DAL.
Okay. So let's look up threonine, THR. So yes, they're all the same first two letters, right? They're all THR is all AC, 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 AC. So could any point mutation here still produce threonine? No, no. Where would the point mutation have to occur?
The last one.
Right, so no point mutation. And it would be the same thing here. Would be silent. Right, because same thing's going to be with aspartic acid, ASP. GAU or GAC, right? If I change that letter, it's not going to be aspartic acid anymore.
But sometimes it can, right? Like with, there's argons on there 2 times. Like you
Arginine is CGU, CGC, CGA, CGG, and then AGA, AGG. So yeah, that would be an exception. And then there's another one that's like that too. I thought. Yep, leucine's on there twice. But that would only be the first or the second letter for that. So because no point mutation at either one of those would be silent, let's say instead of at 5 and 8, let's do 6 and 9. Could a point mutation, that was a G, let me rewrite that. Could a point mutation occur at 6 and 9 and be silent? Yes. So if we are going to have a point mutation on number 6, and it still needs to be a threonine. So threonine is coded by ACU, ACC, ACA, and ACG. So what do you want to change this to? You can change it to anything you want, right? You could put anything there. If you make that point mutation now, let's see, let's just make it a C. If that becomes TGC, that would become what up here? ACG. And is that still threonine? Yes. Okay, what about for aspartic acid? There are only 2 that code for aspartic acid, right? So what point mutation here would still give you aspartic acid? I have GAU, so what's the only other way that I could still have aspartic acid? Right, if this is GAC, right? So in other words, if this was CTG, that's the only point mutation that would still be silent. Does this one make sense on how you do it? Okay, good. So now let's just talk about 6 and C and D. Could the same polypeptide be produced if an insertion mutation occurred anywhere in the DNA? Doesn't really matter where. Would it be exactly the same as, oops, where's my hand? As this? If I had an insertion mutation anywhere. What does an insertion mutation do?
Oh, no, it changes.
Right, it changes the reading frame. If you're reading in triplets and I have an insertion mutation here, right, I'm now reading a different frame. And so every single amino acid is going to be different.
I was thinking like it was just like the silent one, just somewhere.
Oh, okay. No way is the answer. Right? Because inserting a new base would shift the reading frame. And everything after, all are different. right? So let's pretend the insertion occurred right here. You might have the same first amino acid, but everything after that's going to be different. Could this same polypeptide be produced if a deletion mutation occurred anywhere? No, no way. Again, what's the same reasoning, right? No, because remove a base equals frame shift, right? So again, everything after that would be different. So if you lost this U, or excuse me, if you lost that A, instead of reading AAA, it's now reading AAT, right? Then GTC, then TAC, then AC, right? So everything's going to be different after that. So no, insertion and deletions are going to shift the whole reading frame. You're not going to get the same result. Silent mutation, depending on where it occurs, right? When we saw with 5 and 8, those wouldn't be silent mutations, right? But if you saw it looked at 6 and 9, that would be silent. We understand how the silent mutations, insertion and deletion mutations affect the polypeptide working backwards.
Yeah.
All right. How do we feel about this? Feeling good?
The only one I missed was whenever I looked at, I thought it was talking about something else.
Oh, okay. That's just interpreting the question. All right. Well, then I will stop.